So today, thank you for the invitation, and today I want to talk about some of our preclinical work which we've done to look at the impact of nicotine in brain development. How do you get it to advance? Oh, this one.
After that, it should work there.
Should work there?
Yeah, here or there.
Okay. So this basically shows the slow decline of cigarette use among teens over the last few years. By 2009, however, 20% of 12th graders still smoked cigarettes. This has changed very dramatically in the last couple of years. This shows a decline in cigarette use between 2011 and 2014. So this is high schooler use of cigarettes. It's declined very markedly. And in its place, we have a huge increase in e-cigarette use or vaping. So it actually tripled in the last year alone, according to government data. Why? So there was actually an interesting LA Times article that looked at this just after those data were released. And they interviewed high schoolers in Long Beach. And Dean Wilson, an 18-year-old who attends Polytechnic High School, said that though he tried regular cigarettes, he preferred vaping with e-cigarettes because they're new and trendy, because they come in a variety of appealing flavors. Apparently, there's at least 7,500 flavorants that are now added to e-cigarettes, something that is illegal for cigarettes, but not for vaping. He also believes they're less dangerous than traditional cigarettes, and they're supposed to have less nicotine. Fellow student Jose Sanchez, who actually didn't vape or smoke because he was an asthmatic, but he noted that they're a lot more popular now and people want to fit in. Even public health professionals are celebrating the use of the switch from cigarettes to vaping in teenagers. Dr. Michael Siegel, a professor at Boston University School of Public Health, was interviewed for the article and was quoted as saying the CDC should be celebrating that we're seeing a decline in youth smoking. So is vaping really safer than cigarettes is a question that is important now, is an important public health policy question. So the primary ingredient in e-cigarettes is nicotine. So unlike tobacco smoke, which contains over 8,000 constituents, e-cigarette smoke is cleaner. It still has other additives, but really nicotine is the primary ingredient. However, nicotine has been shown to impact developmental plasticity, and it basically interacts with nicotinic acetylcholine receptors in the brain and the periphery. And those, we and others have done a lot of work to show that those receptors have critical developmental roles. And that if you interfere with those receptors, if you activate them during development at the wrong time, you can completely screw up development, basically. So we were interested to know, and we started this work before the vaping craze began, but it's certainly relevant. We were interested to know what the impact of nicotine was on adolescent brain development itself. Now, adolescent brains, for those of you who either have been adolescents or have had adolescent children, we all know that the adolescent brain is a little different from the adult brain. We have birds and the bees lobe and memory for music lobe and all the answers and so on and so forth. So definitely a little different. They may look like adults, but they don't always think like adults or act like adults. And part of the reason for that is adolescence is a period of really active brain development. There's a very active myelination and synaptic pruning and functional connectivity that's occurring in the brain at this time. And it's particularly evident in the limbic system and the prefrontal cortical areas. These areas control cognition and emotional response. So these are actively developing. as are some brain systems that are neurochemical systems that are critically important in cognition and emotional response. The dopamine system, for example, is in the adolescent brain is very immature and has a very distinct functional role from that in the adult. And we and others are showing now that the serotonin system, which is one of the earliest developing systems in the brain, it previously was thought to be mature by adolescence, is in fact anything but mature by adolescence. So our hypothesis is that nicotine may impact these systems and in adolescent brain and could therefore produce a long-term change in brain and behavior. Now, we can't do this in humans. It would be completely unethical to start giving nicotine to teenagers and seeing what it did to their brains. So we use we use rodents. And here we basically compare the maturation of the rodents with that of humans. And so brain maturation in both humans and rodents is actually a long period, and it's not just defined by puberty, as many people think. It actually starts before puberty in both humans and rodents. But as you can see, there's a period that strongly overlaps with puberty. So we, in our rodent studies, started looking at about postnatal day 28, which is considered to be very early adolescence and would be equivalent to about 12 years old in humans. One of the primary behavioral models that we use is intravenous self-administration. We basically put a rat in an operant chamber. The rat has previously been catheterized with an intravenous catheter that is connected to a computer that will deliver drug. The chamber has two either holes or levers that the animal can interact with. And it has a light that when the light is on, it tells the animal that drug is available for them to take. There are two, in this particular chamber that's shown here, there are two holes, but often it's levers. One of the holes, if the animal pokes their nose in the hole, it delivers drug, and a light goes on, the cue light goes on. The other hole, there are no programmed consequences, but we monitor activity in that hole because it's a way of looking at locomotor activity. So is the animal taking drug just because it's more active, or does it prefer the hole that delivers drug? So when we look at drug intake in adolescents as compared to adults, we find that there is a significantly higher intake of nicotine, especially at higher doses in adolescents than in adults. In adults, there are strong negative aversive effects that occur at higher doses, but those are not evident in adolescents. So they're capable of taking more drug without those aversive consequences. But most of the work that we've done is actually similar to the work that was presented by Andrea earlier from San Bernardino, asking the question, does early exposure to nicotine, early adolescent exposure to nicotine, does it change brain and behavior? And this graph shows the likelihood, and although it's a fairly old graph, the data are still relevant today. The likelihood of a teenager using other drugs depending on whether or not they smoked. So the green bars are smokers and the blue bars are non-smokers. And you can see that smokers are far more likely to take other drugs. They're more likely to use cocaine, they're more likely to do binge drinking and heavily drink if they also smoke. Now there are two schools of thought as to why you see this concordance. One is that social influences, and that these are just bad kids, basically. Bad kids will do bad things, they'll smoke, they'll drink, they'll do all sorts of things, and they'll hang out with other bad kids who encourage them to do the same thing. However, Denise Kandel, who's married to a Nobel Prize-winning neurobiologist, was probably influenced by him. She's an epidemiologist that demonstrated that there was progression in drug use in teenagers from smoking and alcohol in early teens to marijuana use to then heroin, cocaine, and methamphetamine use in older teenagers. And she proposed that there was actually a sensitization that occurred. So it was a neurobiological mechanism. The adolescent nicotine use then sensitized the brain to other drugs. So basically, that's what we tested. We tested whether adolescent rats who smoked, or in our case, who were given nicotine, were then more likely to abuse cocaine. And so we used the approaches that I showed. We used self-administration as one way of determining whether the animals actually liked cocaine better, found it more reinforcing and rewarding. So our experimental design was one in which we delivered drug by intravenous administration. So we find our adolescent rats to be very, very stress sensitive, and they really don't like injections. And so when we gave multiple injections, we were getting stress effects rather than drug effects. Besides which, if we gave the drug intravenously, it better modeled the pharmacokinetic disposition of smoking than giving drug by other routes of administration. So we would basically catheterize the animals and then give them 3 days or so to recover. And then at postnatal day 28, we would give a daily, one injection, actually 2 injections, 1 minute apart of nicotine, and we would do that for 4 days. And what we were trying to do was to model early smoking. So whereas other models, the one that Andrea presented earlier, for example, really look at chronic effects, what we were interested in was early effects. When teenagers smoke, they usually smoke at home between leaving school and between their parents getting home. It's just a couple of cigarettes. And so we basically used doses of drugs that produced blood levels that were equivalent to 1 to 2 cigarettes in humans. And we did that 4 days in a row. That was it. We then looked, and we always did everything in parallel. We used adults that were 3 months old, and we did exactly the same experiments in parallel. Following day, we then took the animals and did neurochemical analysis or behavioral analysis. And in some cases, we left them for 10 days and did some behavioral tests. So first, we used CFOS messenger RNA expression. This is an autoradiograph that shows distribution of CFOS, which is an immediate early gene that is expressed when a neuron is activated. So we use that as a readout to look at neuronal activation. And we looked at brain areas that have been implicated in addiction. And we found that nicotine in adolescence, and it's this very low dose of nicotine that we gave, 60 micrograms per kilogram, tiny dose, that it selectively activated both the basolateral amygdala and the nucleus accumbens shell, 2 of the parts of the circuit in adolescents but not adults. So in this graph, white bars are animals that got saline, yellow bars are those that got nicotine. And just one very low dose of nicotine selectively activated the reward mechanism in adolescents but not adults. We then went and looked at monoamine levels and monoamine transporter levels in these brain areas. So we looked at dopamine, the transmitter, serotonin, the transmitter, and also transporters for those 2 transmitters, dopamine transporter and serotonin transporter. And we looked in prefrontal cortex, basolateral amygdala, nucleus accumbens, all of which are part of the addiction circuitry. And then dorsal striatum, which is less involved, at least in initial acquisition of drug use. The pluses show where there was a significant difference between adolescents and adults, and the adolescents were always lower. So it basically shows that these animals are still maturing and that adult levels are higher than adolescent levels. And when you see the asterisks, that's where there was a significant nicotine effect on the neurochemical composition. So the asterisks are all related to serotonin, which kind of surprised us. We thought that if we were going to make animals more sensitive to drug addiction. It would impact the dopamine system, but we were finding it was impacting, at least presynaptically, the serotonin system. However, when we looked at dopamine receptors, we didn't see any change in binding of any dopamine receptors, and this is shown here for the D2 receptor. Again, white is saline-treated, red is nicotine-treated, and this is in the nucleus accumbens, two subregions of the nucleus accumbens, and in the dorsal striatum. However, when we looked at receptor activation with a biochemical assay that allowed us to actually look at the endpoint of receptor activation, we found that there were significant increases in the activity of the dopamine receptor in some of these rare areas. So although the dopamine presynaptic system wasn't affected, the D2 receptor function itself was. And D2 receptor is one of the dopamine receptors that really has completely different functions in adolescents versus adults. So then we looked at behaviors that might depend on the D2 receptor. And so here we're showing actually a time course of 30 minutes after administration of either saline, which I don't know if you can see this. It's basically just the squares on along the x-axis, so our animals just didn't move after they got saline. This dotted line, black dotted line. Okay, just use it. Great. So the black dotted line is animals that got quinpirole after saline pretreatment, and this red line is quinpirole after 4 days of nicotine pretreatment. And we saw a great enhancement of this D2 receptor-mediated locomotor response. When we gave the same drug the same time, so from postnatal day 28 to 31, and tested the animals 10 days later on postnatal day 41, this locomotor response to quinpirole is an age-dependent one, and it's disappearing by postnatal day 41, but we're still seeing the enhancement by the nicotine pretreatment, even though it was given 10 days earlier. On the other hand, if we gave the nicotine just the 4 days before postnatal day 41, we didn't see that enhancement. So this effect of nicotine is specific for early adolescence. P41 is still adolescent. It's around about the time of puberty, in fact, but we don't see the effect anymore. And we didn't see an effect in adults. We were interested to know whether serotonin somehow produced this effect on dopamine receptors, since we've seen big changes in serotonin. And the bottom line is that this is nicotine's enhancement of quinpirole-induced locomotion in the absence and the presence of a serotonin 1A receptor antagonist. So this effect was completely blocked by a serotonin 1A receptor antagonist. We then looked at locomotor sensitization, which is considered to be a model of addiction. And so we looked at giving several days of cocaine treatment. And if you look in the adult, every day the animal got cocaine, they moved more. We then took a day off and challenged with cocaine, and you can't see it down here, but the animals who got saline pretreatment, their cocaine challenge is down here, and this is the motor response of animals that had the 3 days of prior cocaine exposure. That's called locomotor sensitization. We didn't see that in adolescents that had been pretreated with saline. On the other hand, adolescents that had been pretreated with nicotine, we saw very beautiful locomotor sensitization.
I want to block your little thing here.
Oops. Thank you.
Still here, right?
There we are.
Okay.
Yeah. So as I said, we're seeing locomotor sensitization. Some people believe that that is a more direct way of looking at addiction circuitry, and that this would predict that we'd actually see an enhanced rewarding or reinforcing response to cocaine. So we did that experiment next. And as you may be able to guess, once I find these were the animals. So this is cocaine at self-administration. over 5 days. So this is the first day they were just put in the box and allowed to figure out which hole delivered cocaine. And very quickly, the animals that had received the nicotine pretreatment understood that they were getting cocaine, whereas those that adolescents received saline pretreatment or either of the adult groups, they didn't respond on as much. And this effect was seen on day 1, which is really pretty astonishing. Animals usually need to learn a task, but they immediately knew that this was cocaine and that they liked it. So everything else that we did in this study was just, we looked at that first day of acquisition. Again, this is data showing that the serotonin 1A antagonist blocks the effect. So this again is an effect that's mediated by serotonin 1A and receptors. So the model we basically developed is that nicotine is releasing serotonin, which is activating serotonin 1A receptors. We showed that these were postsynaptic receptors, and somehow changing dopamine D2 receptor signaling to cause cocaine self-administration and to enhance quinpirole locomotion. We then also showed that it wasn't specific to cocaine. So this is methamphetamine. Here we're showing reinforced versus non-reinforced responding. And the saline-pretreated animals on day 1 didn't show any difference, but the nicotine-pretreated animals immediately recognized that one hole delivered methamphetamine and they liked it better. We didn't see that difference in adults. Alcohol, we saw the same thing, but not in adults. So one of my students said, but people don't smoke first and then drink. You know, alcohol and tobacco are 2 of the most co-abused drugs. Why don't we try them together? So we did. So we basically did an experiment in which we again compared adolescents and adults. We looked at males and females, although I will be showing females here. We looked at alcohol, nicotine, and alcohol plus nicotine alone. We basically did exactly what I said before. We catheterized the animals, let them recover for a few days, and then we did an escalating paradigm because they don't like very high doses of alcohol to begin with. So we did 3 days of a low dose of alcohol or nicotine or the combination. Then we escalated to mid dose, and then we went to a high dose. And you can probably guess which is the group, without even looking at the legend, which is the group that received the combination. This is alcohol intake, and the animals that received them both took much more alcohol. In fact, these are the adolescent males that after a 1-hour self-administration period, they were drunk when they came out. They couldn't stand straight. It's very hard to get animals to self-administer alcohol to the point of drunkenness. The other 3 groups are alcohol alone, which is the blue, and then adults that received either alcohol or the combination of alcohol and nicotine. So this is a very specific effect for adolescent males. Females, interestingly, didn't show this, that nicotine is enhancing alcohol intake. We were then interested to know, are there long-term effects of this self-administration of drugs? So we did exactly the same, except we let the animals at the end of the self-administration period, we let them rest for a week, and then we did a drinking in the dark paradigm, in which we gave them 2 bottles, one with water and one with alcohol, and we allowed them to drink from either. And then again, we escalated, starting at a low dose of alcohol and then going to a high dose. And then finally doing a 2-hour drinking in the dark to see, that really forced them to decide did they want the alcohol or not. And these are the results. This is what I've shown you already, the self-administration. The adolescents that got nicotine along with the alcohol took in much more alcohol than either alcohol alone or either of the adult groups. But interestingly, when we looked at how much alcohol they took in the drinking in the dark paradigm, the animals that got the combination, which is this red group, were enhanced as compared to those that had previously self-administered saline, but no more enhanced than nicotine alone. So they'd never seen alcohol. They had only seen nicotine, and that enhanced alcohol drinking. So in the adolescents, it appears to be nicotine exposure that is increasing long-term alcohol preference. In the adults, we saw something different. The nicotine and the nicotine plus alcohol did not affect the preference, but prior alcohol self-administration did. So again, we're seeing drug-specific effects depending on the age. Finally, we've shown now that there's an effect on adolescent brain and behavior, but of course, many young women who smoke or vape are going to expose their fetuses to the drug as well. And so we've done quite a bit of work to determine whether that prenatal exposure to nicotine impacts brain and behavior. And these are autoradiograms where blue, it goes from blue to red, where red is the highest binding, of nicotinic receptor distribution in adult on the, here's the adult, and then 3 days before, gestational day 19 is 3 days before birth in a rat. And you can see that there's already a lot of nicotinic receptors in fetal brain, and that we have data that shows those receptors are active. So does activation of those receptors by nicotine actually produce long-term changes? So we addressed that by giving nicotine, and this time we did do a chronic administration via mini pump from gestational day 4 through to, the pump actually stopped delivering nicotine at about gestational day 20, and the animals were born gestational day 21. So they're exposed to nicotine through all the major phases of brain development. At birth, the animals were switched to a surrogate dam, so the dam's behavior as a result of prior nicotine exposure wasn't affected. And then at postnatal day 21, we weaned the animals, separated them by sex, and then we tested them behaviorally and neurochemically as adolescents on postnatal day 30. On this graph, we're looking at preference for sucrose. So this is natural reward. We had shown, I didn't show the data, but adolescent nicotine doesn't affect natural reward. It's really specific for cocaine. But prenatal nicotine does affect natural reward. And pink are the animals that were prenatally exposed to nicotine. Turquoise were the saline control. So it actually decreased the intake of sucrose pellets, decreased natural reward. And at high doses of cocaine, it greatly increased cocaine intake as compared to animals that had been exposed to saline. We were interested to know, we used the CFOS approach again to determine whether cocaine was doing something different in the brains of adolescents who had been prenatally exposed to nicotine versus saline. And so we treated animals with saline or cocaine, looked at CFOS expression, and then we compared effects depending on whether animals had received gestational nicotine or saline. Looking at the nucleus accumbens, we basically found, again, pink are prenatally exposed to nicotine. We found a non-significant, but an increase in the neuronal activation of a shell by the highest dose of cocaine. It was a significant increase in the activation of the core by the highest dose of cocaine. And then the dorsal striatum, both, it wasn't dependent on whether the animal had been prenatally exposed to drug. Cocaine activated that part, that region in both groups. But the important thing is that the nucleus accumbens is the output system for motivated behavior and for addiction and reward. And so it's being selectively impacted by the prenatal nicotine. So to briefly conclude, nicotine produces long-term changes in developing brain, both adolescent and fetal. Among the many nicotine-induced changes that are seen, we've shown alterations in dopamine system activation, which leads to enhanced sensitivity to abused drugs. And as a conclusion, e-cigarettes are not as safe as they seem. So let's hope that the senator is successful in his legislation. And I just want to thank the many people in my lab and the funders.
Great talk, thank you. I'll start with the first question about the prenatal nicotine decreasing sucrose intake. Is that good? Maybe we should be giving our mother nicotine, or do you want to explain that?
From a metabolic perspective, that might be the case, but actually sucrose intake, decreased sucrose intake is an animal model for depression. So it may basically show that there's emotional dysregulation.
Questions? Sure does. Questions? Okay, thank you very much, and thanks for coming a fairly long distance.